cell communication. Very short, okay? I'm not gonna go too crazy here. Cell communication, okay? So a cell is trying to communicate with another cell. How does it work? That's what this is. Like signal, okay? So this entire thing, I mean, is a very, I mean, again, I'm trying to put it into a 30-minute, like, explanation here, okay? But it's if you guys go to school, maybe a little deeper dive is needed for this, okay? So, so best example I can give you guys, for example, is a cell within the adrenal gland. So adrenal gland, okay, I mean, they got cells in there, okay? So for example, guys, there is right now, guys, there's a bear running towards you right now, okay? You guys accidentally, there's someone let the bear or whatever, let loose, and they're running towards you, okay? So your eyes are sensing, okay? So sensing what? Okay, so there's a bear running towards you. They're sending signal to the adrenal gland. Okay, like I mean, for guys, right? Like for guys, right? I mean, like I know it's COVID times, right? But I'm sure you guys got into like some sort of an argument or fight. Okay, so like oh yeah, come at me, bro. What happens, guys? Your heart starts bumping, like beating faster, and then like what the hell happens? Like other stuff happens, like your eye dilates, like oh come at me, bro. Like come, like wait. You gotta understand, guys. When people drive, like dude, when you honk, dude, this is a life lesson. When you honk, dude, Americans, like, I don't know, they take this very personally. Like, when you honk, fuck you, man, like, come out. Yeah, like, so okay, so which means, well, yeah, adrenal gland is what? Like, they're the ones that send the signal down. What? Okay, like, dude, you're about to go into a fight. Send the signal to what? Other cells. Okay. So typically, adrenal gland is what? Send the that's what epinephrine, right? Epinephrine. So there, like this is a hormone, by the way. So this is basically it's a hormone. I mean, people think like, oh my god, it's nothing fancy. It's just it's a chemical, like molecule. It's a molecule, okay? So epinephrine. So they release a molecule called epinephrine. So again, the target cells. These are what we call the target cells, right? A target cell must respond. So what's a response? Like heart cell is yeah, they gotta start beating faster. Okay, so. And also what? There's the liver cells are supposed to what? Produce more glucose, okay? So because what? You need more ATP, right? So they're supposed to produce more glucose. So they did test on this, by the way. So they did a test on this. I mean, by the way, but before that, so the communication, something like these are what they call a signaling molecule. In bio, we call this what? Ligands. Ligands, okay? So whatever the messenger, in this entire thing is what? Yeah, they're ligands, right? So basically what happens is, if you look at it, so like the target cell. So if this is a liver cell, okay, this is like in the textbook, okay? If this is a liver cell, so the response to the epinephrine for the liver says what? Yeah, dude, they store the sugar as glycogen, right? And we'll convert this to what? Glucose. Okay? Depolymerization of what? Glycogen to glucose. So epinephrine, okay, activates the enzyme, okay, which converts this into glucose and what? Put into a bloodstream and what? Yeah, the bloodstream and what? The cells will take this and what? Get ready for the fight, right? So if you look at it, guys, right? So basically what they did, okay, basically the scientists did is this. This is what they did. I'm not kidding. In a beaker, okay, they put glycogen, they put the enzyme. So, I mean, dude, we talked about it, right? Enzyme is, must be, like, in bio, right? Enzyme must be activated. Okay, enzyme is not gonna just oh yeah, enzyme glycogen, oh yeah, turns to glucose. It doesn't happen. So enzyme must be activated. Usually what, like phosphorylate the process, right? So they maybe put some ATPs here, like whatever. Okay, ADP, ATP, whatever. Okay, and then what? Okay, so if they start to put in epinephrine into this container. You know what I'm saying? So yeah, dude. If they have so basically look at this, okay? Epinephrine activates this enzyme, okay, from which converts the glycogen into glucose, right? So, do you think it's gonna work? This is just a beaker with water, glycogen, enzyme, and ATP, okay? And what? They put what? Epinephrine. Do you think this enzyme will start to break down the glycogen, guys? It doesn't work. They noticed the what? It's not working. So it doesn't matter how many epinephrine you put in, how much enzyme you have, how much ATP you got, how much glycogen you got. It's not working. Okay? So which means what? Something is happening in the cell's cytoplasm. Okay? Something is happening there. Okay? So they probably convert the epinephrine into something else. That's the understanding of this process. Okay? That's basically what this chapter is about. So this chapter is very simple. Okay? This chapter is absolutely like, it's hard chapter, but AP Bio, they don't go too far. Okay? And like we talked about the Olympia, like that we talked about, they barely have one question here, by the way. I don't know why, I have freaking no clue, okay? So let's take a look at it, guys, okay? So, like, a little more specifics now. So, this is what we call, guys, cell communication, which is broken down to 3 parts, okay? Reception, okay? Signal transduction, and response. Okay. So this is basically, guys, the 3 phases of the cell communication. Okay. And you just have to know 2 receptions for this chapter. 2 receptions. Okay. So right now, guys, I mean, epinephrine is a polar substance. Okay? Epinephrine is a polar substance. Okay. So this is a cell. This is a liver cell. So epinephrine is a ligand from adrenal gland. They sent it, okay? So, like, okay, so we gotta think about receptor for this molecule, okay? So keep in mind, guys, because this is a hydrophilic, ligand, they are not able to come in, okay, into the cytoplasm. Which means what? They need to have a receptor must be on the cell membrane. So remember, guys, the beaker. Think about it, guys. Beaker with the enzyme and glycogen. Okay, you gotta understand, guys. This guy never comes into the cell. Okay, so what? So the enzyme is saying, like, who are you? Okay, I don't know who you are. Okay, so it's a messenger. So which means what? They never enter the cell. So which means what? They are bound to a protein receptor. Okay, on the cell membrane. Because why? This is a polar ligand. Okay, it's a polar ligand. So once the receptor is activated, once this guy's activated, what happens, guys? Okay, so dude, I tell you guys, okay, for AP Bio, you need to know this specific, like the receptor, which is called a G protein coupled receptor. Okay, G protein coupled receptor. They love the term coupled in bio, by the way. They love the term. I don't know why. I have freaking no clue. Okay. Yeah, oh yeah, coupled. Yeah, they work together. That's why, probably. Okay. And then so this is called G protein. Okay. Nothing fancy. You might have to explain this on your AP, by the way. Okay. So once the receptor is activated by binding to a ligand, okay, then the G protein gets activated. So this G protein is called G protein. Why? They have a GDP, okay, like the guanosine diphosphate bound to them, okay, like ADP, like ATP, right? So now, once this is activated, they convert this to GTP, which is what? This is now an activated form of G protein, okay? So now, dude, think about it, guys. They have to tell the enzyme to what? Okay, so dude, you gotta break down the glycogen, okay? How are they going to send the message? They have to produce what? Something we call a secondary messenger, okay? They call what? Secondary messenger. And typically, guys, in bio, in AP Bio, secondary messenger is what, guys? Typically, cyclic AMP. Cyclic AMP is what we call the secondary messenger, okay? So now, there's an enzyme next to the G protein. Dude, I tell you, you gotta know this, okay? Adenylyl cyclase. I tell you guys, your teacher will definitely talk about it. You gotta know it. Okay, I don't know what school you go. Guys, I mean, I forgot most of the Jericho, if I remember correctly. They will talk about it, adenylyl cyclase. So the G protein will activate this guy, okay, and what? Convert ATP into what? Cyclic AMP. Yeah. So in biochem chapter, we talked about what AMP is. We talked about AMP was, right? AMP is what? So ATP is what, guys? Adenosine triphosphate. Okay? What's AMP? Adenosine monophosphate. Okay? So what is this guy doing? What are they doing, guys? They're chopping off 2 phosphates. Okay? So you got 3 phosphates. Okay? And then what? They're chopping these out. Okay, now it's what? Your monophosphate. So, you know, what do we call this, guys? Pyrophosphate. Okay, pyrophosphate. That's what 2-phosphate means. Okay, it turns into what? AMP. So cyclic AMP, guys. I mean, I'm not gonna do crazy. Cyclic AMP, guys. Okay, so which means this guy is now bound to the 3rd carbon here. So it's a cycle here. Okay, it produces a little little ring here. That's called cyclic AMP. So cyclic AMP is known to be what we call they call this what? Secondary messenger. So which means what? Instead of what? Instead of the epinephrine, it's what? Cyclic AMP is now the new messenger for the cell. Okay? And this just like what I just mentioned right now, that's the reception. Okay? So the receptor activates by the ligand. Okay? It activates the G protein, which swaps the GDP into GTP, activates adenylyl cyclase, which will what? Convert the ATP to cyclic AMP, which is what we call, guys, what? What we call this, what, guys? Secondary messenger. So it's what? The process converts what? Epinephrine to cyclic AMP as the messenger. That's what happens during the reception portion of the cell communication. Okay? Any questions? Let me know. Try to listen very carefully now. What happens after this? So you have to ask yourself, guys, okay? So remember the beaker, right? Remember the beaker? We put a glycogen, we put the enzyme, they'll break down the glycogen into glucose. So if I add cyclic AMP, right? Would they break down the glucose? Still no. No, still not. So what the hell happens then? This happens. Signal transduction happens, okay? Signal transduction pathway. I guarantee you guys this is on your AP, okay? It's good to know. And I tell you guys, whatever I'm talking right now, guys, is basically what I mean. Hopefully, your teacher teaches, okay? And this is the most that you need for AP, okay? They don't have a lot of questions on this one, but they will have. I mean, they don't have a lot, but they will have questions in it. So what happens? And one word should pop up, okay? When you hear this, like, you gotta understand, guys. Cascade. You gotta understand cascade, okay? It's not like detergent, okay? So the cascade of phosphorylation. That's what happens, okay? So whenever they say signal transduction pathway in the cell communication, is what? Cascade of phosphorylation. That's something you have to know. So what the hell is this? So what happens, like, so this, you might say, like, what the hell, why do they do this, okay? So basically what happens is, the cyclic AMP, okay, so will activate a protein called kinase, okay? This is kinase. So there's a kinase, okay? So you guys should know at this point what kinase, what they're doing. They phosphorylate the other parties, okay? They activate the other guy by phosphorylating, okay? Cyclic AMP will activate, so this is inactive right now, okay? There's the first kinase right here. Kinase A, they call it kinase A, kinase B, kinase C, whatever, okay? So this guy would what? Now, inactive form right now, but due to what? The secondary messenger from the reception is what? This guy will now be activated, okay? Which will what? Yeah, this guy, being activated, what? Uses ATP, okay? This guy will, the cascade, which is what? Yeah, this guy will activate the next guy, kinase number 2. Okay, this is kinase 2, okay? Kinase 2 is inactive right now, okay? But due to the kinase 1, what happens, guys? This will be now, this guy, the kinase 2, is now phosphorylated, because what? ATP, right? ATP is now turned into what? ADP, and the phosphate here, okay? So kinase 1 is what? Activating kinase 2, okay? Because what? Their job is take the phosphate from here and put it on this guy, okay? Inactive, now they're active. Kinase 2 is now active. You understand, guys? And then what do you think these guys will do? What do you think? This same thing, okay? Next kinase. Kinase 3, okay? Inactive. And using ATP, what happens, guys? It will activate into what? Kinase 3 phosphorylate, now active, okay? And that's what they do. Constantly uses ATP for this, okay? And once, like, so that's, I mean, so it goes like, you know, it's like multiple of these steps. That's what they call by what? Cascade of phosphorylation, okay? So, and then also, guys, I mean, so yeah, once, like, keep in mind, guys, once they've done their job, because cyclic AMP, guys, okay, so yeah, they activate this guy, what happens, guys? They turn this into what? Inactive form, which is AMP. There's an enzyme called phosphatase, phosphodiesterase that will turn this into that. Inactive, they get rid of this, and what? So now also what? Once they activate the next step, they go back to inactive form. There's a phosphatase that take this out, you know, so that's what happens, okay? So if you look at it, guys, so this is what they call, okay, cascade of phosphorylation. That's what we call, guys, signal transduction. And if you ask yourself, guys, like, what the hell, why? Why do you think they're doing this very long process? Okay, what the hell? Like, okay, so just send this to the enzyme to activate it. What? Like, yeah, just let's get it going, right? Why they're doing this, guys? What do you think? It's for the main reason is what? Amplification. So that's the main reason. Amplification of the signal. So what it means is, yeah, one single epinephrine, okay, can produce what? Thousands of cyclic AMP will produced. Okay, it's not only, I'm only showing you one, okay? Yeah, one of this can produce what? Like 100 of the cyclic AMP, okay? And 100 cyclic AMP can what? Activate 1,000 kinase 1, okay? And 1,000 kinase 1 can activate what, guys? Like 10,000 kinase 2 molecules. So, which means the number amplifies as you go down. So that what? Okay, so which means it will activate, so they're not activating one enzyme, they're activating what? Thousands of the enzymes, okay? To what? Break down the glycogen. Dude, you're about to get into fight. You can't just have one glucose, okay? You gotta have what? Like, dude, yeah, keep in mind. Yeah, 1 mole of glucose is what? How many glucose molecules is this, guys? 6 times 10 to the power of 23 glucose molecules for 1 mole, okay? Which can produce what? Like, what do they say, guys? Like 700 calories, kilocalorie or something? Okay? Just so you know. Just to let you know, if you run around 1 mile, you burn around 100 calories, kilocalories. Just so you know, if you run 1 mile, okay? So, which means like, yeah, if you run, this is like around energy of what? Running 7 miles, because you know. So, which means like, okay, so you need like a lot of glucose molecules, which means what? Like, dude, yeah. One enzyme activated. Oh yeah, I gotta break this down. I'm gonna break this down. It takes a lot of time, which means what? You gotta activate many of these guys. Okay, so that's what the amplification is. Okay, and then also what? Like, dude, there's a lot of glycogen and what? Like, you need a lot of these guys. That's why they have to amplify the signal. Okay, that's just one epinephrine can what? Activate what? Thousands of these guys. Okay, that's the purpose of what? Amplification. And there's a different reason also. Okay. So the different reason is, a different reason for this process, like many different steps, is what? Liver cells, they have a different purpose for epinephrine. Liver cells what? What do they do? They have to produce glucose, right? But now, heart cell, what's their response for epinephrine? Do they produce glucose, guys? No. Their purpose what, guys? Starts to beat faster. They have a different response. Which means what? This portion, which is a signal transduction, is what, guys? Yeah, depending upon what cell you are, they have a different response. Which means they have what? A different transduction process. Okay, so you know. So, which means what? Yeah, the different cells respond to what? Same molecule, okay, same epinephrine. Okay, even the ligand is same, they respond differently. Which is what? Their transduction signaling is different within the cell. Okay. So yeah, amplification, yeah, correct. But they have what? This can also what? Decide what kind of response you're gonna get. Okay. And let's take a look at, guys, the response. Let's take a look at this guy. So how does this work? So I tell you guys, response can be like, dude, anything. Okay. Response of the cell signal can be anything. It could be what? It could be activation of the enzyme. Okay. Just like what we're talking right now. It could be also what? It could be also production of enzyme. Okay. It could be also what? Yeah, deactivation of the enzyme. Right. It could also be what? Yeah, dude, like, okay, so open up a, like, you know, the channel for the mid-cell membrane, okay? Like, it could be what? Like, it could be many things, okay? But now let's take a look at just what we're talking about now. So, so you look at it, guys. Okay, so yeah, let's go into kinase, okay? So now let's talk about kinase 5. Let's went into like, okay, so they got into a 5th kinase. So take a look at this. So this is the enzyme that will break down, this is the enzyme that breaks down the glycogen into glucose, okay? And now they have what? Activated kinase 5. This is now activated. Do you understand, guys? Yeah, this guy is now active. Based on the cascade of phosphorylation, you got what? Like, dude, like 100,000 of these guys, okay? 100,000 of these guys are now activated, okay? What do you think this guy will do? What do you think this guy will do? What do you think, guys? This guy is what? Will use ATP and what? Activate the enzyme, which will break down the glycogen. Okay? Oh yeah, use ATP and what? Phosphorylates the enzyme. Okay, now this guy's active. Okay, that's the job of what? Enzyme called kinase. Okay, which means what? Yeah, they uses ATP and what? Activates the enzyme which is inactive. What? And now this guy will be what? Able to break down the glycogen into glucose. Okay? So just lay it out. That's basically how things occur inside the cell. Okay? That's the reason why if you throw in epinephrine, if you throw in the enzyme and glycogen, it's not working. Because why? They forgot this step. Okay? This is the guy that's responsible for activation of what? The enzyme that will break down the glycogen into glucose. Okay? And that's what we call, guys, a signal transduction pathway. You do not have to know a lot for this chapter, okay? But you might want to read the chapter, have an understanding of this, okay? What it is. Sometimes, guys, by the way, you know, like, yeah, this is simple and plain simple, whatever, okay? One other thing that you have to know for this chapter is this. What if for example, guys, okay? Like there's a question that we just did, okay? In the cell organelle question, like chapter. Yeah, what if it's an estrogen? If estrogen is a ligand, okay? So again, yeah, before this, right? Before like AP Bio way back, okay, when I was in AP Bio back in 2003, 2003. Wow, that's like what, 20 years ago? Yeah, so 2003 AP Bio, okay? Yeah, you had to memorize estrogen's polarity. Meaning like estrogen is what? Oh yeah, this is what? Non-polar ligand, okay? Yeah, what do you think will happen? This guy will what? Come into the cell cytosol, okay? Cell cytoplasm. Which means what? There must be a receptor molecule. This is AP, okay? Receptor protein is inside the cell. So this guy right here is what, guys? It's inside the cell. Receptor is inside the cell. So typically what happens, guys, receptor is outside for what? Like polar ligands, right? But now receptor is inside the cell. So this is where the AP, they want you to definitely know this, guys, where the receptor is inside the cell, which is what, yeah, the reception occurs in the cytoplasm. Okay? And typically, right, just like now, this one, there's no, so not every, like, you know, communication result in like cascade of phosphorylation. This does not go into phosphorylation process, because you know, this will directly activate like in an enzyme. Okay, well this guy will go into a nucleus and what? Tell the nucleus to what? Produce a certain protein. So yeah, so this will act as a transcription factor and what? Tell the nucleus, okay, so give me this protein. Okay, so just like what we talked about, like albumin or something. That's one of the stuff that we talked about. So So this guy, so it depends on what kind of ligand it is, okay? They go into a different reception and also what? Different what? Like, yeah, response. They might not go into that transduction process because, you know, any questions, guys?
So certain types of them go on the cytoplasm rather than a receptor.
Yeah, because what? They just come into a cell. So if you have a receptor on the cell membrane for this guy, I mean, it doesn't matter because what? They just go in. Alright guys, so I'll assign guys photosynthesis homework, okay? And also cell communication, that's the last one by the way, okay? So, and then we'll discuss this a little more, photosynthesis guys a little more, and then the last chapter we'll discuss next week will be the cell cycle, okay? That's the last chapter. Cell cycle guys, I mean, that will be discussed like your school is in December, okay?